B lymphocytes, and humoral immunity. B cells are responsible for defending the body against extracellular pathogens by producing antibodies for humoral immunity. They recognize antigens using B cell receptors, also known as BCRs. They differentiate into plasma cells and secrete specific antibodies and form memory B cells for long-term immunity. And the antibodies help by neutralizing toxins and viruses, enhance phagocytosis by opsonizing bacteria. It activates the complement system because the antibodies bind to the antigens, and helps with agglutenating pathogens by the cells clumping together. And then some types of the B cells are plasma cells, memory B cells, and naive B cells. And the plasma cells are antibody-producing effector cells. The memory B cells are long-lived cells for rapid secondary response. And naive B cells are mature B cells not yet exposed to the antigens.
T cells are a type of white blood cell that are part of the adaptive immune system, meaning they help the body recognize specific pathogens and respond to them in a targeted way. They get the name from the thymus, which is the organ where they develop and mature. Unlike B cells, T cells do not produce antibodies. Instead, their main role is to help control immune responses and, in some cases, directly kill infected or abnormal cells. This makes them especially important for fighting viruses because viruses hide inside our own cells and for recognizing cancer cells. There are main three types of T cells that are important to understand. First are helper T cells, also called CD4 positive T cells. These are basically the coordinators of the immune response. They release chemical signals called cytokines that activate and guide other immune cells, including cytotoxic T cells and macrophages. Next are cytotoxic T cells, also known as CD8 positive T cells. These are sometimes called killer T cells because their main job is to directly destroy virus-infected cells and cancer cells. They do this by triggering a process called apoptosis, or programmed cell death, which safely removes damaged or dangerous cells. Finally, there are regulatory T cells. These help keep the immune system under control, limiting immune responses and preventing the immune system from overreacting or attacking the body's own tissues. So overall, T cells are essential because they help coordinate immune responses and directly eliminate infected or abnormal cells. Cell-mediated immunity is part of the adaptive immune system, and unlike antibody-based immunity, it relies on immune systems themselves rather than antibodies to fight infections. This type of immunity is especially important for dealing with viruses, some intracellular bacteria, cancer cells, and even transplanted organs. Several different immune cells work together in cell-mediated immunity. Helper T cells coordinate the response. Cytotoxic T cells do most of the direct killing. Macrophages help by engulfing pathogens and presenting antigens. And natural killer, or NK cells, can also destroy abnormal or infected cells, especially early in an infection. The process usually starts with a macrophage or another antigen-presenting cell that has engulfed a pathogen. This cell breaks the pathogen down and displays a piece of it, called an antigen, on its surface. A helper T cell recognizes this antigen and becomes activated. Once activated, the helper T cell releases cytokines, which are chemical signals that stimulate other immune cells. These cytokines activate cytotoxic T cells and help increase the overall immune response. The cytotoxic T cells then go out into the body and look for the cells that are infected or abnormal and displaying the same antigen. When they find one, they bind to it and release substances that trigger apoptosis, or programmed cell death, which destroys the infected or cancerous cells in a controlled way. At the same time, macrophages continue to help by engulfing pathogens and cleaning up debris, and NK cells also kill abnormal cells without needing prior activation. Clinically, this process is very important. For example, HIV attacks helper T cells, which weakens cell-mediated immunity. This type of immune response is also involved in organ transplant rejection and is used in some cancer immunotherapy treatments. So overall, cell-mediated immunity is a coordinated, cell-based defense system that protects the body by finding and eliminating infected or abnormal cells.
The nature of antibodies. Antibodies are produced by plasma cells, and they are Y-shaped glycoproteins where each antibody has a specific region that binds on particular antigens, and it acts as a lock and key. It also binds specific antigens such as proteins, polysaccharides, nucleic acids, and lipids, and it neutralizes pathogens directly. The basic structure is 2 heavy chains and 2 light chains. The variable region is antigen binding. The constant region is immune activation, and it is linked by disulfide bonds. The key properties are specificity, which binds one antigen, diversity, which is millions of variations, and memory response, which is faster on re-exposure. And then there are 5 main types, and that will be the IgG, the IgA, the IgM, the IgE, and the IgD.
Clonal selection theory explains how the adaptive immune system responds to a unlimited range of antigens using a pre-existing population of lymphocytes. Each lymphocyte recognizes a specific epitope on an antigen. Naive lymphocytes have antigen-specific receptors but are not yet activated. When they encounter their matching antigen, they receive signals that activate them. Activated lymphocytes proliferate rapidly through clonal expansion, generating many identical copies. This creates effector cells that combat the antigen and memory cells for faster responses in the future. The process applies to both B cells and T cells. B cells use B cell receptors, while T cells use T cell receptors. Effector cells include plasma cells that produce antibodies and cytotoxic T cells.
For this slide, I'm going to explain T cell development, which is the key part of lymphocyte development. T cells start in the bone marrow, but they migrate to the thymus to fully mature. The thymus provides the environment needed for proper development and selection. One of the most important steps during maturation is T cell receptor or TCR gene rearrangement. This process creates a unique receptor on each T cell, allowing the immune system to recognize a wide variety of antigens. In the thymus, developing T cells first enter the double negative stage, where they do not express CD4 or CD8. As they continue developing, they become double positive. This stage prepares them for selection process. Selection is critical for self-tolerance. During positive selection in the thymic cortex, T cells must be able to recognize self-MHC molecules. If they cannot recognize them at all, they die because they would not function properly in the immune system. After that, negative selection occurs in the thymus. In this step, T cells that bind too strongly to self-antigens are eliminated to prevent autoimmunity. Together, these processes ensure that surviving T cells can recognize antigens appropriately without attacking the body's own tissues. Finally, once they pass selection, these cells leave the thymus as mature but antigen inexperienced, or naive T cells. They then circulate through lymphoid organs ready to respond when they encounter their specific antigen. Overall, T cell development ensures both diversity in antigen recognition and self-tolerance, which are essential for a properly functioning immune system. In this slide, I'll be connecting the Christian worldview to the immune system as a whole and just bacteria and different organisms in our lives. First, in Genesis 1:27, it says that humans are made in the image of God. From a Christian perspective, this gives inherent value and dignity to every human life. When we study immune cells like T lymphocytes, we're really studying part of the intricate system that protects that God-given life. Next, in Psalm 139:14, it says, "We are fearfully and wonderfully made." When we look at the complexity of the immune system, it really reflects an incredible level of organization and detail. From a Christian worldview, this complexity can be seen as evidence of intentional design. Studying immunology allows us to better understand that design and apply it in ways that promote healing, disease prevention, and medical advancements. Finally, Christians believe that God created everything good. However, the fall introduced brokenness into the world. This includes disease, pathogens, and death. From this perspective, illness is a part of the fallen world. But studying microbiology and immunology becomes a way to respond to that brokenness. Healthcare and scientific research can be viewed as participating in restoration. Overall, a Christian worldview encourages us to value human life, appreciate the complexity of the immune system, and use scientific knowledge responsibly to care for others.